Hi, everybody. Welcome to this session of Expert Research Talks. For those watching at home, welcome. And please, we're going to have our first presenter come up, and then we can pause to take questions before we go on to the next presenter. And if you're watching from home, please submit your questions through the app. Well, actually, I forget where it is. I think it's intuitive where it is, but I will find your questions and we'll go through them after this first presenter. So I'd like to welcome Dr. Gрэм Коллингридж.
Thank you very much. So this is a fantastic conference. I've really enjoyed it up until now. So if there's any organizers present, brilliant. Just honored to be able to present here. I'm going to present some data which is unpublished. We're planning to write it up shortly. So looking forward to getting some feedback, critical or otherwise. But I thought I'd also start by just introducing myself and how I got into the Grin field. So I wanted to be a footballer. That's soccer in American. But I realized even my dog was better than me. But I was at the University of Bristol, where on the faculty in the Department of Pharmacology was Jeff Watkins, who is the father of the glutamate receptor field. I mean, he's done more than anybody to identify AMP receptors, kainate receptors, and of course NMDAR receptors. So I became inspired to work on NMDAR receptors and have been doing so for my whole career. But in terms of Grin, my involvement in the Grin project started on this date, and I know this date because it's a date that England qualified for the semi-final of the World Cup. This is in Toronto. There's a load of England supporters like me. Everybody was drinking vast quantities of alcohol, except me. I was drinking vast quantities of coffee because in the afternoon, Amy Ramsey had invited me to talk to a small NMDA receptor group organized by Keith MacArthur. And this was when I met Keith and his wonderful son, Bryson. And he said to me, is there anything as an NMDA receptor researcher we could perhaps do? And what I'd realized moving to Toronto is that they had a fantastic mouse facility. So I said, well, we'll try and make the mouse with the same variant as Bryson. And I said, we could probably make a second one as well. So he suggested I made this second variant, the Center for Phenogenomics, which is a Mount Sinai and Sick Kids run facility. So we made these two mouse models. And so what I'm going to do is give a presentation of our phenotypic characterization that's been going on now for a few years. So just a bit recap, they're both in the Grin1 gene. They're both glycine to arginine mutations in different locations. So the G6, it's too difficult for me to control this. The G620R, so if anybody went to Jinyol's poster or his brief presentation yesterday, I'll be presenting some of that data on the left-hand side, and I'll be showing for the first time the comparison with the G827R on the right-hand side. And what I can say is because these animals come from the same facility, all the experiments have been done in the same lab by the same researchers, I think you can confidently say that if there are similarities, are genuine similarities, and differences are genuine differences. It's not some kind of cross-lab effect because of different levels of stress or animal housing, etc. So we know quite a bit about these receptors when they've been expressed in recombinant systems, mainly from Steve Trinellis' lab and a few others around the world. So I've listed these references here. What we knew is that these have a profound effect on the NMDA receptor, but what we wanted to know is what does this translate to in the native environment of a synapse in the mammalian system. So the first thing is that the mice were born fine. The 827s were more difficult, but we're now getting them breeding pretty well. We never see any homozygous mice, presumably because it's embryonic lethal. The hets are a bit smaller in both cases, and they're lighter than their wild type counterparts. And I'm not going to present much behavioral data today, but in addition to reduced body size and weight, they have locomotor deficits, increased startle response, increased tolerance to thermal pain. Well, this is the 620Rs. We haven't done much behavioral characterization of the 827s yet. And a few other learning deficits that Jinyol mentioned yesterday. In terms of the gross brain anatomy at the level we've looked at, which are just initial stains, there's no obvious difference. Cell counts are about the same. So this is the first perhaps surprise, because we know from a lot of literature that the NMDA receptor is very important in development, but we don't see any obvious gross abnormalities suggesting that the one good copy is sufficient for the general wiring of the brain. So the electrophysiological characterization has been performed by Patrick Tidball, who's done most of the field potential recording, and Junior has done most of the patch clamp recording. So the standard protocol that we used, I will eventually learn how to get this to work, is to record field excitatory postsynaptic potentials from the Shafer-Clattrell common serial pathway. These are blocked by AMP receptor antagonists, so this AMP receptor mediated synaptic transmission. We can block AMP receptor mediated synaptic transmission with MBQX, lower the magnesium concentration, and record NMDA receptor mediated synaptic responses, which are blocked by AP5. And so this way we can look at AMP receptor mediated synaptic transmission and NMDA receptor mediated synaptic transmission directly. And for the wholesale recording, again, looking at AMP and NMDA receptor currents and spontaneous and miniature evoked synaptic currents. So the first thing is we looked at the size of the synaptic responses, starting with the presynaptic fiber volley, which is this tiny little component here, which is the axons coming down, or the action potentials coming down the axons. And if we plot the amplitude of the fiber volley against the stimulus intensity, then there's no difference, suggesting that on average, you've got the same amount of connectivity in the HETs as you have in the wild types. And the same is true for the 827Rs as well. So again, this is consistent with no obvious gross abnormalities. And again, it's suggesting that the development has gone, is fine, it's normal. In terms of AMP receptor mediated synaptic transmission, this is also normal in the 620Rs. So here what we're doing is we're plotting the size of the slope of the field excitatory postsynaptic potential as a function of fiber volley amplitude. As you can see, they're superimposable. So AMP transmission is also normal in this mouse. The first surprise, however, is in the 827Rs, the AMP receptor mediated synaptic transmission is actually enhanced slightly, but highly significantly. So you've actually got more AMP receptor mediated synaptic transmission in the HETs than you'd expect. In terms of the underlying mechanism for that, we looked at pair pulse facilitation, which is an index of presynaptic release probability. And there was no difference here in the 620Rs. But in the 827Rs, there's a change in the pair pulse facilitation, which is consistent with an increase in the probability of release, suggesting that in this mouse model, the AMP receptor mediated transmission is increased because of an increase in the probability of release. And that is confirmed by looking at the spontaneous and miniature responses. No difference in the 620Rs, and an increase in the mini frequency in the 827Rs. So this was the first surprise, and that is AMP receptor mediated transmission is actually enhanced in this mouse model. But what of course we're really interested in is NMDA receptors, because this is variants are within the NMDA receptor. So in the 620R, there is a substantial reduction in NMDA receptor mediated synaptic transmission. As you can see, down by over 50%. And in the 827Rs, it's an even greater reduction in NMDA receptor mediated synaptic transmission, down to about 75% reduction. So this is exactly what you'd expect. And we assume that the increase in AMP is some form of compensation to counteract this dramatic reduction in NMDA receptor mediated synaptic transmission. So one of the properties have been identified by Steve Trenelis in the recombinant systems is there's a change in magnesium sensitivity in the 620Rs. So we assumed that we might see something similar if we looked at the synaptic responses. But indeed, what we found was that we couldn't detect a change in magnesium sensitivity of these receptors when they're at the surface of the membrane, assuming they are making it to the surface of the membrane. So this is looking at the NMDA receptor-mediated synaptic response with increasing concentrations of magnesium. So as you increase the magnesium concentration, you've got more and more of the response. And this is a superimposition of the wild type and the hets. And you can see there's really no difference in their magnesium sensitivity. And we did the same experiments with the 827Rs. And again, there was no obvious difference in the magnesium sensitivities. We suggest that in the native environment, they're behaving differently, or perhaps they're not actually making it to the surface of the membrane, which is what we assumed was the case for a while. And another way of looking at this was to look at the current voltage response by isolating the NMDA currents and measuring the NMDA receptor synaptic currents at different membrane potentials. And there are slight differences, but nothing to really suggest that there's anything major going on here. So we'll conclude that actually the voltage dependence of the magnesium block is unaltered in these mice. The other next surprising observation, however, is that when we look more carefully at the kinetics of the synaptic currents, there was a small reduction in the decay kinetics in the 620Rs. Now, I have to admit that I didn't believe this for a long time, and Junior would know that he's trying to convince me this is a real observation. I said, no, it's got to be a space clamp artifact, because we're trying to clamp these cells with massive dendrites. But eventually he convinced me it's real, and one of the reasons I now believe it is that when we look at the 827R, even though this is a more dramatic reduction in the NMDA response, there's essentially no change in the kinetics. So of course, what we've done here is we've peak scaled these responses. This will be a lot smaller than the wild type, but you can see that the decay kinetics are essentially identical. Whereas here, there's a small but highly significant difference in the kinetics of response. And you might wonder, why is that important? That's important because it's telling us that the receptors are probably getting to the surface and they're having different properties. Whereas this one, it could be due to the fact that it's a trafficking deficit. So one possibility we imagined is what could be possible is that with a reduction in the GluN1 subunit, you're getting a different assembly of subunits at the surface, and that could explain the kinetic difference. So very recently, we've looked at some experiments. This is using field potential recording again, where we looked at the sensitivity of MVP, which is a GluN1 2A, more selective, whereas Rho at this concentration is very selective for GluN1 2Bs. And there is a small sensitivity in the wild type, suggesting that some of the receptors here are heterodimers, whereas the vast majority are probably heterotrimers. And we're seeing a small but significant difference in Rho sensitivity. So this would be consistent with the wild type receptors having more GluN2B diheteromeric assemblies, and then in the knockouts, this is reduced, and therefore we're having more of the triheteromerics with the same sensitivity to NVP. Right. Finally, the thing that I'm most interested in, which is synaptic plasticity. So because NMDA receptors are profoundly affected, one would predict, or one would hope, or I would certainly hope, that if you have less NMDA receptor activation, you have less LTP. And indeed, that's what we found. So in the 620Rs, the LTP is reduced by about 50%. And in the 827Rs, where the inhibition of the NMDA response is even greater, the LTP is practically abolished. So there is a profound reduction in synaptic plasticity in these mice. And that is widely believed that this plasticity underlies learning and memory and other cognitive deficits. So I would predict that this is the reason that we get cognitive problems in patients who have these same variants. Okay. So we're at the point now of wondering whether what we're seeing is less native receptors because the mutant receptors are not being assembled or they're not being trafficked properly, or whether we're getting expression of the variants, but they're less effective when they're in the synapse. And a clue to that may be the case is the alteration in the kinetics of the response. So again, very recently, what we've been doing is doing some biochemistry. So what we're showing here is the total expression in the wild type and the hets of the GLUN1 subunit. And this is the surface of the synaptosome preparation using biotinylation. And you can see essentially there's no difference in the total expression of the synaptic surface expression of the 620Rs. In contrast, when we look at the 827Rs, again, there's no difference in the total expression of the GLUN1 subunits, but the surface expression is dramatically reduced, around about 50% reduction in surface expression. So what we think is happening is that in the 620Rs, we're getting expression of receptors which comprise some regular wild types, but also some with the subunit with the variant in. And these are generating a smaller current with different kinetics due to the slightly difference in the subunit assembly. Whereas in the 827Rs, it's more straightforward. We're not getting assembly of receptors. We're not getting to the surface. And so what we're seeing is the residual response from the wild type receptors. So in summary, we can conclude that the 620Rs and the 827R Grin mouse models have substantially reduced NMDAR receptor dependent synaptic transmission and LTP. The effect is larger in the 827R than the 620R. And we believe that different underlying mechanisms responsible for the reduction in NMDAR receptor function in these two mouse models. So in the 620R, we think it's due to the presence of variant subunits at the surface. Whereas with the 827R, we're pretty convinced that there's actually a reduction in the number of receptors expressed. So it's just the wild type receptors that are being expressed. We don't see any significant impact on gross brain anatomy, cortical number, or connectivity. I'm not saying there aren't subtle differences. We haven't looked in great detail, but superficially at least, the brains seem okay. And that's, I think, really, really good news, because if the brains developed okay, and we can acutely rescue the NMDAR receptor phenotype, either with drugs or with gene replacement, then I'd be optimistic that we should get a fairly good recovery of at least some function. And there was a compensatory mechanism we picked up in the 827R mice that was not seen in the 620s. And I think that's because of the profound reduction in NMDAR receptor mediated transmission has led to some compensatory mechanism leading to an increase in the probability of release of glutamate to increase the AMPA receptor mediated synaptic transmission. Okay, so I've gone through that quicker than I anticipated. So it just remains for me to acknowledge that the work has all been done at the Lunenfeld-Tanban Research Institute at Mount Sinai Hospital. Also, the animals are generated at TCP, which is the building next door. I also have a position at the University of Toronto. The work that I've described, Jiniol, who presented the poster and sitting there, will answer all the difficult questions. And along with Patrick, has done all the electrophysiology. Yesel and Shinwon did some behavior. I haven't had time to go through the behavior, but that's still ongoing. Jonathan Thacker did the biochemistry at the end. Thursday did the anatomy. John has been looking after the project. And my picture's in black and white because it's such an old picture. I've been involved in the background. So thank you very much.
If you'd like the questions to be picked up on your recording.
Hi, nice talk. I was wondering if I'm picking up on this kinetics of the decay, the faster kinetics. Have you looked at what happens in current clamp and integration towards action potential? Does the fact?
Okay, so in terms of the kinetics, it was all done under voltage clamp. It's the only way we can really get an accurate measurement of the kinetics.
So my question is really, have you tried current clamp?
I don't think we have. Not that I'm aware of. No. I'm totally on top of what's going on, as you can tell. But with the field potential recording, of course, that is giving you an estimation of what's happening, a population of neurons which have not been clamped. But we don't see it. I mean, the transmission looks pretty normal, apart from the reduction in NMDAR receptor-mediated transmission. We don't see any hint of loss of inhibition. We don't see any hint of epileptiform activity.
So we can discuss afterwards, but in Dravet mouse model, what we've seen is that the EPSP duration affects spike timing, and that can, on top of everything else, can also relate to information processing and deficits there.
Yeah, so which mouse was that again?
And Dravet syndrome mouse model, so
Okay.
not green.
Yeah. There's a question there.
Thank you. That was very nice work. Questions are one, why is it that these two missense variants are so different at an electrophysiological level?
Yeah.
And two, does that correlate with clinical phenotype in any way?
Okay, so good questions. Well, I'm sure the answer to that is the region of the receptor that's been affected, because although it's the same amino acid substitution, they're different regions of the NMDAR receptor. So one is critically involved in the assembly, and that's why we don't think we're getting assembly of the receptors in the 827s. Sorry, what's the second question?
Does this correlate with the clinical phenotype in the patient?
Yeah, as far as we know, I mean, it's obviously difficult to do a direct comparison between a mouse and a human being. But say, for example, the body weight is consistent with my understanding of what happens to the small number of patients with that same variant. The hypertonia, which I think we're going to pick up with the locomotor problems, is consistent. Intellectual deficits are consistent. So as far as we can tell, there's a lot of similarities. I think the thermal sensitivity is being picked up as well as a experience in the children. But we're in the process of compiling an up-to-date list of the human phenotypes, thanks to Tim. So we'll be able to answer that question more thoroughly.
Because the practical implication is, if there's a way to predict the phenotypic severity based on genotype, that would be incredibly helpful, especially for missense variants, because we see a patient with that. Yeah.
We was talked yesterday, I think, about a program trying to predict the outcome based on the amino acid substitution, which was running at about 80 to 90% accuracy, I believe. But ultimately, I think you have to do the experiment in the mouse model. I mean, Steve will correct me if I'm wrong, but I think in some of your studies, the initial prediction was the 620 was a gain of function because of reduced magnesium sensitivity in the recombinant system. Is that right?
That was right until we did the biochemical surface.
Yeah. 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 Okay. So, yeah. So ultimately, whilst the recombinant system is really good for getting an accurate estimation of what's happening, ultimately, I think it has to be done in the mouse as well to know to study the receptor in its native environment. So I think I would really need to do a different mouse for each variant. And each one of these runs at about $500,000 to $1 million in terms of making them, maintaining them, paying for people to do the experiments, et cetera, et cetera.
Going to be less technical, I suspect, but because you talked about, or maybe the question won't make sense, but this presence of the variant subunits in the G620R.
Yeah.
So when we think about what that means, of course, then as parents, we jump to, so what does that mean in terms of, would you want to reduce that expression? Like, are there things that we can start to consider now as a result of knowing that?
Yeah, I mean, I think it's important to know that to design the right approach. It's more complicated if you've got variants expressed at the synapse. If you've just got a knockdown, then an increase in the number of the native receptors would be the logical way to go. Probably putting in more native receptors would work as well, because the more native receptors, the greater probability of obtaining native receptors at the synapse. but there's always going to be that competition of the express variant receptors. So ultimately, we have to try, and we will try. I mean, what is good about this is the phenotypes are so clear. I mean, they are massive reductions, and they're very consistent. So if it's possible to rescue, we should be able to see it.
Thank you. And thank you for all the work that you've done and to your team as well. So, Danielle.
Right. So I don't think I saw it. When you do the biotinylation to look at the surface expression and you see the trafficking defect, did you do 2A and 2B as well?
I was dreading you asking me a biotinylation question.
This is a nice one.
But I can
have
answer that. I can
I have
answer
some
that
other
one.
ones, right?
We haven't yet, but we're going to, obviously, because it would be really nice to see if there is a subunit switch or change.
Well, because it seems like a profound deficit, right? So I mean, and if GLUN one's not on the surface, then nothing is. I mean, that deficit should be.
Yeah, so it should definitely go down where the GLUN1 goes down, but in the one where the GLUN1 doesn't change,
Yeah. Right.
our prediction is that there may be less B. So it's going to be very small. So whether we'll pick it up or not, I don't know, to explain the kinetic shift. But we'll try. And if we struggle, I'll come. I'll say help.